Great. So I guess we get started. So yeah, my name is Vikram. I'm a PhD student at Johns Hopkins University, working with Dr. Ben Langmead. And I'll be talking about a project that we call Sigmoni, which is applying kind of compressed text indexing to the problem of nanopore signal classification. Yeah, so just a quick overview about nanopore sequencing, as I'm sure some of you might be familiar with it already. We can essentially sequence DNA and RNA molecules with nanopore sequencing by passing these molecules through an artificial nanopore. What a lot of people might not know is that the raw output of the nanopore sequencer is this electrical signal that I've shown here. And what this is essentially a continuous measurement of the electrical current through this nanopore, because we pass a voltage across the nanopore, and the nucleic acid has a characteristic resistance, which causes a change in the current that we can measure out over time as the sequence passes through the pore. And so the standard pipeline is to pass this raw electrical signal into a base caller. These are generally neural networks. Recurrent neural networks are the state of the art for this task. And these can reconstruct the original long reads that represent the underlying sequence that was passing through the nanopore at any given time from this electrical signal. And so with these long reads, you can pass them into any of your favorite downstream analyses pipelines. In this case, I'm just showing an example where you can use Minimap2 to align the reads to on-target or off-target genomes. And so one thing you might want to do is filter out the off-target reads that align to the off-target genomes and keep the on-target for downstream analysis. But a very unique aspect of nanopore sequencing is that you can actually do these decisions in real time with something called adaptive sampling. And the way this works is that as the read is being sequenced in longer and longer prefixes, as it passes through the pore, you can make a real-time decision and tell the pore to eject the read by reversing the voltage that's passing across the nanopore. And doing this in real time, you can save a lot of time by only sequencing a small part of a read that might be in an off-target region and focus the sequencing resources and time and cost on sequencing on-target regions. And so one of the first tools to do this was ReadFish, which uses Minimap, and the signal as it's being output is base called, passed to Minimap, and aligned to your genome of interest. And if it aligns properly, then it continues sequencing. And if it aligns to an off-target region, it can send that reject signal to the sequencer. Likewise, it can continue sequencing the on-target reads. And so this is the overall pipeline, but what a lot of people have noticed is that the base calling step represents a large bottleneck in this pipeline. So this is a previous result that shows that more than 80% of the pipeline for, I think, SARS-CoV-2 variant calling, which started from nanopore sequencing all the way down to the variant calling step, within that pipeline, the vast majority of the compute time was being used on the base calling step. And using a neural network base caller can often be very difficult without hardware acceleration, other considerations to take into account. And so there have been previous research to try and develop tools that can do classification on this raw electrical signal, bypassing the base caller, but still making rapid decisions for this adaptive sampling protocol. So Uncalled was one of the first papers to do this, and how it works is it identifies likely K-mer sequences from the electrical signal and then uses an FM index to align these K-mer sequences to your genome of interest and then make a decision accordingly. SigMap is another tool that does this as well, and it uses a KD tree as its underlying data structure, but it works in a very similar way to digest the electrical signal into seeds and then query those against a reference. But one thing we noticed is that both the FM index and KD tree and these other data structures that have been used to work with this electrical signal and query it to these genomes, they often don't scale with complex repetitive genomes or large collections of genomes, such as a pangenome. So one thing that we wanted to address was, can we build a nanopore raw electrical signal classifier, but using an underlying pangenome index, allowing adaptive sampling to scale to larger and larger references. And so one pangenome index is the R index, which we've shown, colleagues in our group have shown with a tool called Sigmoni that uses the R index to classify the base call nanopore reads. And it showed that the R index can compute exact matches between a query and a reference very quickly for these large collections of reference sequences like a pangenome, and it can classify these nanopore reads very well. So what is the R index? It's sort of made up loosely of three major components. The first component being the run length encoded Burrows-Wheeler transform, the Burrows-Wheeler transform being sort of the core of the FM index, but in this case it's run length encoded, which saves a lot of space because in these pangenomes and repetitive text, the BWT tends to have a lot of runs of similar characters that can be compressed very well. It also contains a subsampled suffix array, subsampled at the run boundaries of the BWT, as well as some auxiliary data structures that allow us to compute exact matches very efficiently. But the key is that all of these data structures are sort of big O R, loosely big O of R in size, and that's really important for being amenable to a pangenome context. And so the Sigmoni framework also showed that you can compute these exact matches in linear time, and I'll show how that works against these large pangenome reference index. And so just an aside on big O of R data structures and why they're very useful in this day and age, these are just examples, and I guess the emojis are slightly different on a Microsoft computer, but these are some examples of pangenomes across the tree of life that have been published in the last year or two of tens to hundreds of reference quality assemblies. And I'm showing here the compression ratio, which is N, the size of the references if you concatenate them back to back, and R, which is the number of runs in the BWT of the reference. And you can see that the ratio R grows far smaller than N because of all the inherent repetition between similar sequences in a pangenome. And so it's very ripe for compressibility and working in this big O of R compressed space. And so scaling analyses like adaptive sampling and reclassification to work in this big O of R space is crucial in the pangenome era. So going back to the R-index for exact matching, I'll show an example of how the Sigmoni framework allows linear time exact match computation. And what it does is it computes matching statistics, which are a form of a maximal exact match. In particular, they're a half mem, so they're right extendable. And I'll show an example of what that means in a moment. And so we have an example query and a reference sequence here. And so at every position in the query, we can compute what is the longest right maximal match that appears in the reference. So at the first position, AT appears in the reference, but ATG doesn't. So we can extend that match by 1. And so the longest match would be of length 2. Likewise, TG doesn't appear in this reference. And so the half mem at that position would be 1. And we can step through and we can compute this. And the Sigmoni framework can use the R-index to compute this very efficiently in linear time with respect to the query, which is very useful. And Sigmoni showed that we can do this in base called nanopore reads. And so it does this by computing something that we call pseudo-matching lengths, which are very similar to what I just showed, those half mems, but they're faster to compute in a streaming manner, which is very important when you're streaming in reads in real time. So we developed Sigmoni, which is similar to Sigmoni, but as you can probably guess, it adapts the Sigmoni framework to work with nanopore signal. And so it's a signal-based reclassification system that uses the R-index. And the way it works is conceptually, it quantizes this continuous electrical signal that we get from the nanopore sequencer into a discrete alphabet space. And then it projects the reference into the same alphabet. And then once we have these two texts in some arbitrary alphabet space, we can compute exact matches as we would with any sort of text with the R-index Sigmoni framework. And so similar to Sigmoni, it can classify nanopore reads, but it avoids that base calling step. And it does this directly from the signal by using this rapid binning procedure, which is sort of akin to a very naive base caller. And so how does that work? So here's an example of a signal, and I've sort of drawn it in the y-axis here is the picoamp, so that the current, and we're sort of measuring it over time. What we can do is draw these sort of arbitrary boundaries, which we call bins, and then quantize this continuous signal into a sequence of these bin characters, and they're arbitrary. In this case, we've chosen 5 bins. And so we can treat the reference the same way, and we do that by digesting the reference into k-mers. In this case, a 6-mer, because a 6-mer is what's characteristic of the electrical signal, because a 6-mer is what fits in the pore at any given moment. And so we can compute the current that we would expect to get if we were to sequence in an ideal world this reference from start to finish. And then we can do the same quantization procedure on that to get the expected bin sequence of the reference. And now we have these two kind of discrete alphabet texts, and we can compute our exact matches with the R-index. And so just as an aside, we can deal with sort of signal processing issues like the segmentation errors that we get using homopolymer compression and other text-based techniques. And so, like I said, we can just replace these bins with really any alphabet. The alphabet's sort of arbitrary as long as it's sort of the number of bins is fixed. So in this case, I just pick A, B, C, D, E, but it doesn't really matter. It could be any characters. But essentially, the query is now in the same alphabet space as the reference, and we can compute exact matches as we would any text. And so the way this works is, I hope the cursor's not blocking this. Yes. So the way this works is I'm showing, it's kind of small on the screen, but the Burrows-Wheeler matrix and the Burrows-Wheeler transform along with the suffix array of just an example text, which is made up of a concatenation of a bunch of different reference sequences. So the way the R-index works is that it subsamples the BWT and the suffix array at these runs of the BWT. So we save a lot of space there, and then the number of runs grows sort of sublinearly relative to n. But also, Sigmoni introduced this idea of a document array, where each of the suffixes in the Burrows-Wheeler matrix is sort of tagged with the sequence of origin in the underlying pangenome or reference collection. In this case, it's just 3 bacterial species. And so we can tag them with that information. So as we compute matches, we can see where those matches are actually coming from and which reference they're coming from. But that didn't quite cut it with the noisy electrical signal that we're dealing with Sigmoni. So Sigmoni introduces an additional feature, which we call shredded documents. It's essentially a document array, but rather than associating every suffix array entry with a reference in the collection, we're now associated with the specific region within a single reference in the region. So we sort of shredded each genome into sort of 1 KB regions. And so this lets us get a slightly more coarse grain or fine grain location information for our matches, which lets us triangulate the location of the exact matches across the query and make a more accurate decision downstream. So the output of the R-index is sort of the half mems across the query that I showed earlier between the query and the reference. And so using this location information, we can tie it together and see that the matches sort of all cluster around one specific region, in this case in Listeria. And so we can make a decision with high confidence that this read originates from that region in that reference. So I'll get into some results of how Sigmoni works. Let me check on time. Great. So how Sigmoni performs, I'll start with the index space, and this is sort of enabled by this R-index's great compressibility and scalability. In particular, we were able to index over 7,000 bacterial genomes in just under 2.5 gigabytes. This is a bacterial pangenome that represents a mock community, which I'll describe in a little bit. But also you can see the scaling very nicely here when we go from indexing one human genome to a set of 6 sort of reference quality human genomes to all 88 or 89 assemblies in the Human Pangenome Reference Consortium year 1 data set. And as you can see, as the size of the index or size of the reference grows very quickly, the index size sort of grows sublinearly as it scales with R or the number of the amount of unique sequence that's added in with new assemblies. And so you can see the scales very nicely when we compare to 4 other tools, signal-based classification tools. You can see that the only tool that was able to actually index the Human Pangenome Reference Consortium assemblies was RawHash2, which took just over 3.5 terabytes of index size, while Sigmoni was able to index that in just 25 gigabytes. And so that really lets us scale our signal classification pipelines to larger and larger references in the future. And so some results of how it compares to existing signal-based methods in accuracy. So on the bottom here, we're classifying reads, the full read. And so this would be sort of a scenario where you have the electrical signal already sequenced and on your hard drive, but you just want to use a subset of that data for your downstream analysis, and you don't want to run base calling on all of the reads. You just want to take the reads that you know are going to be on target and then run base calling on that. And so in that case, Sigmoni is significantly more accurate on classifying these reads than existing tools. And it can also do multi-class classification. In this case, it's a microbial community where we're trying to classify which bacterial species of origin in this community. And in the bottom case, we're doing a binary classification between fungal versus bacterial reads. You can also see that Sigmoni was the fastest tool to do this full read classification. So if we jump to a host depletion experiment where we're trying to classify reads as being of human origin versus microbial origin, you can see now the index size takes into account that the index includes the human genome, and just indexing the human genome was significantly difficult, or Sigmoni was significantly smaller in index size, and as a result, the time to classification was also much smaller because the index that I was working with was smaller. And so on the top, I'm showing a heat map where we can do multi-classification to classify reads as for a chromosome of origin within the human genome, and on the bottom is the same binary classification scheme where it's more accurate and also faster. And so you can also see that the existing tools like uncalled and rawhash and SIGMAP, SIGMAP and rawhash, uncalled was omitted from this because it couldn't index a human genome, but SIGMAP and rawhash tended to not classify a lot of reads, and we think this is because the index was very large, and so those reads had a lot of seeds that hit to very repetitive portions of the human genome, and so those tools refused to classify the reads. Well, Sigmoni was able to overcome this with the smaller index size and the more streamlined classification process and classify more reads and more accurately. And so in the adaptive sampling context, the way we measured, or way we made this comparison was to classify reads in longer and longer prefixes, the signal at least, where one chunk represents around 400 base pairs worth of sequence. And so you can see that those Sigmoni is not the most accurate in the smaller chunks. As we get to around 10 chunks, it reaches the same level of accuracy while also being significantly faster than those existing methods in the microbial reclassification setting. In the host depletion setting, Sigmoni is orders of magnitude faster than existing methods, partially due to that index size issue that I just mentioned. And the accuracy is very comparable, but there's a slight caveat to this. We found that even though the accuracy is comparable, this was sort of an artifact of the fact that SIGMAP and rawhash didn't classify the vast majority of the reads, and since most of those reads were human, the unclassified in this binary classification were sort of labeled as human origin, so it sort of got them right by default, as opposed to really truly classifying the read as human origin. And so there's a sort of asterisk to the comparability of the accuracy results of this. So in conclusion, Sigmoni is a signal-based classification tool, and it deals with this really difficult problem of classifying signal, and this is because of sort of the noise of this continuous electrical signal. But Sigmoni overcomes this with sort of this novel signal bidding procedure, showing that you can do exact matching even on this continuous signal with the R index, and then adding this approximate location information, which is actually applicable even in the Sigmoni framework of base-called reclassification. And so this shows that compressed indexing is really important and growing in importance as we have scale-level analyses to the pangenome era, potentially improving reference bias in these contaminant filtering tasks. And so here's some QR codes for the proceedings paper, as well as the GitHub repo that has the source code for this for reproducibility. And so with that, I'd like to, if I can go to the next slide, I'd like to thank my lab, my PI, my co-authors in bold, and open it up to any questions.
All right, questions?
Hey, Vikram. Okay, nice talk, thanks. So how practical would it be to do base level mapping, let's say with Sigmoni? And I'm curious whether also would be useful, let's say, to do a two-level approach if it is not practical. Let's say where you first do Sigmoni to get an idea of like what is the region in the reference genome that the read can map to, and then you use the other tools, let's say, to do the base level mapping. So I'm curious about
that. Yeah, I think there might be a hiccup with that with the other methods index size, because you have to you're sort of working with the largest index size of the methods, and that's sort of the bottleneck there. But I definitely think the pre-base calling filtering idea that others have sort of proposed of you have all the signal data just accumulating, but you just want to work with some of them. And so maybe you're waiting for a better and greater base caller to come out in the next couple of years. So you just want to hoard your signal data so that you can get better and more accurate reads. You could definitely just sort of get a rough idea of where it is, then do the base calling, and then you can do like Minimap or something to get a really fine-grained alignment to your reference. I think that's totally feasible and something that I would see Sigmoni doing very well in the future. Yeah.
Thanks.
Hi, thank you for the presentation. I was wondering how easy is it to adapt Sigmoni to a new nanopore chemistry? Because if you have now the new chemistry, you're reading 9 mers, so you're having a larger search space if you convert your reference with the pore model to signals.
Yeah, that's an excellent question. It is not very difficult to move the framework over to that. In that the only thing that really changes is how you sort of treat the reference. And so you have to digest the reference into like 9 mers, like you said, because the 9 mers is now the characteristic K-mer within the pore. And so for that, you would need sort of a pore model that tells you the K-mer, what the expected current is. And there's a a really cool paper that's come out recently from one of the co-authors up here that can generate those as well. And so when we have those, it's pretty trivial to kind of swap in that pore model, digest the reference in that way, and then the signal will be in the same. You can project both of them into that same alphabet space, and it should work very well. But like you said, it does blow up a little bit because the k-mers, there's just more k-mers, there's more 9-mers than 6-mers. And so you'd have to sort of change that binning procedure rather than maybe using 6 bins, which I found works best for R9.1. But if you're going to the R10 chemistry, I found that you have to increase the number of bins. And so that takes a little bit of trial and error, but you wouldn't have to do that per experiment. It's sort of like whenever they release a new chemistry, just sort of adapt the binning scheme and adapt the pore model. And then it sort of works out of the box once you have those set. It's a great question. Thank you.
Interesting. Thank you.